That's a good sign to start as well, I think. So, good morning, everyone. The morning session. Let me start by introducing our first speaker, Professor A.Thielscher. Axel obtained his PhD in 2002 from the University of Ulm, and then also in 2008 with first in biomedical engineering and then electrical engineering. He has since then been working as a postdoc at Brown University and also for a long time at Max Planck Institute for biological cybernetics as a researcher and project leader until 2016. But he will also be working since 2012 at the Danish Research Center for Magnetic Resonance and also the Technical University of Denmark, so where he's mainly focused on transcranial brain stimulation. So,
yeah, thank you. Thanks for the kind invitation here. Presentation of my work. So I'll talk about, I have included this brief abstract here instead of a title. I'll talk about volume conductor modeling for applications of in EEG and also brain stimulation. And I have to start newly. Okay, so what is brain stimulation? I mostly talk about two techniques here. One is transcranial magnetic stimulation, and the other one is transcranial electric stimulation. And both use the same principle, namely they use electric currents to change the membrane potential of the nerves, of the brain, and by that modulate neural activity. They just use different physical principles. So this magnetic stimulation uses electromagnetic induction, so that we have just a brief current flow through the wires of the coil, which creates a time-varying magnetic field, which creates then a time-varying electric field in the brain, which then acts on the nerves or the neurons. And here it's pretty obvious, right? We just attach electrodes on the scalp and lead the current through. The principle is the same. There's a big difference in terms of the strength of the electric fields which are induced. Here, we induce fields which are really strong enough to excite nerves. So when we stimulate with TMS, we can induce action potentials. In electric stimulation, usually we have relatively weak currents, and that's just because we not only induce effects in the neurons of the brain, but also in our peripheral neurons. So we feel it, and meaning that it's not dangerous, but when we get too strong, people just say, now it's getting uncomfortable. So that's why we actually limit it to relatively weak currents there, which means that we don't induce action potentials in the brain, but we just have currents which modulate the activity of ongoing neural activity by slightly changing the polarization level of the membranes. I'll also talk about EEG, where, first of all, it's obvious this is a stimulation technique, this is a neuroimaging technique, but the point is that actually on a physical level, they are quite similar. So what you have here in EEG is you have some source here in the brain, which creates a small current, and this is usually pyramidal cell activity, and the current spread out. And as you see nicely here, the current spread out, a bit stronger here, and then they form some kind of pattern. Here, this color change here means that, that's the skull, so most currents don't go through the skull. And then you have some electric potentials which you can record from the skin. And in terms of the underlying physics, which determine how this current is flowing here and how those currents are flowing, that's very, very similar. In both cases, we use so-called volume conductor models, which I'll come back later to. And we have to solve a forward problem, meaning that we know where the currents are generated. And then we determine how the field looks like. Right? The inverse problem in EEG is then often the one which is then used more in practice, where we try to basically get from the distribution of potentials which we have recorded back to the source. That would be the inverse problem. But in order to solve the inverse problem, we need to know the forward problem as well. Yeah? So I have one question for you now, namely, how many of you are familiar with magnetic stimulation or electric stimulation? Should I give a short, so maybe you raise hands when you know a bit about it? Half of it? Okay. So should I give a very short introduction to, okay, it will be only magnetic stimulation. It will be super brief, but let's go for it. So as already said, it's we have this coil. We have a current flow through that coil, which is caused by a stimulator, right? I don't go into the techniques there. But then by that you create a time-varying magnetic field around that coil, which penetrates into the head and there causes an electric field, which excites the neurons. And this was first shown by these gentlemen here in '85, which built the very first TMS machine with this so-called round coil here. And as you see, you need relatively strong currents to create a magnetic field that is then also strong enough to really cause action potentials. So a static magnetic field, by the way, doesn't do that, right? So that's why scanners are safe. You can go in and it's fine. Here we vary this magnetic field quickly in time. So this would be the peak magnetic field. And by that we induce then the electric field by electromagnetic induction. That it works is shown here. This is the very first recording of a finger muscle. This is just an artifact in the recording due to the magnetic stimulus. Because it's so strong, it also induces some artifact in the recordings. And then, as you see, you have to wait for some time, and the time is before you then get a muscle response, so it's switching. And time is just needed because when you induce activity in the motor cortex, where the coil was placed, you just need some conduction time to go down via the nerves to the muscle, and that's what we're seeing here. And with that, you can, for example, now use it in diagnosis to first of all see are these conduction times in a reasonable range, and how strong do you have to stimulate to get such a response, which is, for example, also useful in stroke diagnostics. This is just proof of concept, but where it's actually used most now is in depression treatment, where we use a so-called repetitive TMS protocol. Now, what is that? Repetitive TMS protocols are all protocols where you give not one pulse or a few pulses, but many, many pulses in a certain temporal pattern. I won't go into the specific details of how you then arrange the pulses. The point is that when you give such a protocol, again, maybe for the motor cortex, and you just test how strong you have to stimulate with TMS before you do that, and again after you do that, and you compare both, what you then see is that you can change the excitability of the motor cortex. This is shown here. So this would be the baseline, and the strength of stimulation you need to just induce muscle twitch is here normalized to 1. This would be our RTMS protocols with different types. I don't go into the details. And you see, for example, that this one here, this called ITBS, then increased the excitability, right? So the muscle twitches just got stronger. And this then here was maintained for 20 minutes, maybe it's something, maybe even a bit longer. Now, another protocol here called CTBS magically decreased excitability in that state for some time. When you wait longer, it will level off again. But the point is that you can here induce so-called after effects, which are therapeutically quite interesting. Now, this doesn't help so much when you have an effect which levels off after 1 hour. But what you do then in depression treatment, for example, is that you repeat such a session over weeks. So this is now, the time course here is now weeks suddenly. And there you then start seeing quite stable effects of the treatment. When you treat here, for example, now the typical depression treatment would be that you target the left DLPFC with an excitatory protocol. and then you get relatively good remission rates in patients which are actually a difficult patient group because they're so-called treatment resistant, meaning that they don't respond to medication quite well. And that's where the TMS is so far used most often in treatment. That's quite promising, and we are also still developing further to make this treatment more effective. So what are the problems? The problems are that there's a large individual variability. What this shows here is that for very short protocols, where the textbook would say, well, this type of protocol should decrease excitability. So here, everything should be below 0, and those should be all above 0. This would be a facilitatory protocol. And you see, well, first of all, this was not given strong enough that you have a clear effect there. But even if it would not be that everybody is shifted down. Here, this worked actually quite nice from our TMS researcher perspective. I would say this is a stable outcome here, that we have a facilitation. But as you see that there's, it's a clear, on average, you have a clear effect, but there's also a lot of people here who didn't really respond clearly for that protocol. Then what I just showed you now, that's actually, well, 32% stable decrease. That's good. Obviously, you can try to improve on that. And the error bars here look really small, but the point is that's the confidence of the interval of the mean, meaning that if you would repeat the whole treatment trial, then on average you would expect a curve which lies within that range. The interval variability is much larger, so you have really a lot of responders. You have also a lot of non-responders. So we are really faced with the problem that we have still strong individual variability in our treatment. And we would like to improve on that. And one way of doing this is obviously to personalize the stimulation. And the most straightforward first step one can do is to ask the question, how strong are actually the currents in the brain, and where do they go, and do they go to the same area in all persons with the same strength? And by that, we are now finally at the topic of volume conductor models, where I've been working on quite a bit. So what are volume conductor models? This would be a typical pipeline to build a volume conductor model, a personalized volume conductor model from MR scans, where you start with MR images, which you just segment, so you know then where at which position which type of tissue is. So here would be the eye. Here that's all somehow scalp, a mixture of scalp and muscle. Here you have the scalp, blue is CSF, and then you have the brain, right? So you have segmented it. You have meshed it here. I don't go into that detail. But the point is that once you know where the tissues are, you can also tell basically where in the model you have which type of conductivity. And this is now a coding of the conductivity, where you see that they're quite different. So let's, for example, take the skull again. The conductivity is quite low here, and compared to CSF, which is much higher. And that was also the reason why in the very beginning, there was the shielding effect we had this EEG dipole in the brain, right? And there was then suddenly going the shielding on the outside. That's really the reason because the skull is quite well insulating, right? For example, so that seems to have an effect on how the currents flow. Basically, once you know the conductivities at each position in the head, you can formulate a numerical problem to solve it, solve how the currents are flowing in the brain when you also set the so-called side effect or the boundary conditions, meaning that you tell the program also where actually the currents are flowing into the head. So this would be, for example, a big TDCS electrode represented here, or here in this case, there would be 4 electrodes, and we would have a current flowing into the head here and out of the head here. And once you set that up, you can then solve it as shown here, and you get some visualization or calculation of where the currents are inside the head. Right? That's what volume conductor models are used for. And it doesn't, so the physics are slightly different when we talk about TMS, electric stimulation, or also EEG. But once you have such a model, it's relatively straightforward to solve all three of those, right, problems. So the first thing we did is to ask the question, how much actually does it matter that we know the anatomy here so accurately? Do we need to know really it on a very personal level? And so we just analyzed how sensitive our field simulations are compared to for changes of the anatomy. What we did here is we used the same MR scans, and we ran just different pipelines, which all gave a little bit different segmentations. Maybe most obvious is maybe here this difference where there's just more gray matter. Here it's a bit sparser with more gyri, for example. There's more blue stuff going on here. And we just asked the question, now they're all the same, representing the same head geometry. How much does it matter? And just by visualization of the fields here, this would be for the same electric stimulation montage. So the electrodes are always at the same position and the same amount of current is fed inside. You just see visually, yeah, they seem to differ quite a bit. Maybe that's not such a good thing. We would like to have them really all the same. We then also made a very simple experiment where we split the segmentations up into for example, let's take this. So these are two methods. The names really don't matter, but we split them up into, okay, we sorted them into all the segmentations, all the heads where the segmentations were more similar to each other. This would be this bar, and less similar to each other. This would be this bar. Very simple. And then we just compared how similar are the electric fields. Well, very simple thing is, yes, when you have heads which are more similar, the segmentations are more similar from two segmentations, then also the electric fields are more similar to each other. It's a very simple point to make. So anatomy matters a lot, and when you converge on how you segment the anatomy from the MR scan, then you can also converge on how the electric fields are calculated. So it seems to be important, so we worked a bit further on it. Here, with a method which just segments then, especially the tissues outside the intracranial cavity better, because there are quite good segmentation methods for the brain, right? So free surfer, for example, SPM is also quite good for many purposes. But once you go to a segmentation of the skull and also the scalp tissues, then often what is available or was available for so far is not very stable. So that's why we invested quite some time to just create a segmentation method which works more stably also for the extracranial tissues. And well, this would be then our further previous approach. This is our new approach. What we did here is we compared the simulations, electric field simulations, based on head models created with these two approaches, to electric field simulations based on the ground truth, which was in our case semi-automatically segmented head models where we also had a CT of the subjects available. So that was quite a lot of work to create these head models, but this is as good as we can get when we go manually into the segmentations and do it manually, and also have proper information with CT, for example, which gives very nice information how the skull is really shaped, right? So it's getting quite good when you have proper input scans, I would say. Now, this also matters for the EEG forward program. That's what we started working on more recently with Jesper Nielsen during his PhD. So that would be a typical example on how segmentations of different pipelines can differ, which are available in EEG. That would be our pipeline. And let's compare a little bit. Maybe, yeah, this would be field trip with an SPM segmentation backbone here. Then what you, for example, see is that this blue border would be where CSF and skull meet. And by looking a bit carefully, you see that, well, it's really shifted up here. So this model here would model much more CSF at this position compared to this one, right? So there seem to be very clear differences. Here, M&E is obviously a simpler model. It doesn't have any real information on the differences of the anatomy inside the skull. So it doesn't distinguish between white and gray matter and CSF. It just has these three shells. So there are clear differences in terms of how well the pipelines really segment the anatomy. Does it matter for the forward problem, which is again defined or characterized by placing a dipole at some position in the brain and simulating how the field distribution on the head looks like, and then asking the question here, when we do that for every position here on the head, on the brain, how similar or how different are then the electric field distributions which we're getting on the scalp. And again, compared to a ground truth simulation, which was again informed by our semi-automatic segmentations, where we also had CT in. And there you see that it differs quite a bit, especially here, for example, field trip is not so good here in the orbitofrontal areas, for example. M&E, as expected, has the largest differences because it has the coarsest model, for example, right? So it also matters there quite a bit for the forward problem. So far, I talked about volume conductor models, that the anatomy which you model there really influences how the fields are which you calculate, and that it seems to be somehow important to have a bit more accurate models. Big problem is still that we also would like to validate those models because we don't know, we still have a lot of uncertainties. For example, even when we model the anatomy very accurately, we were not very sure about what is the best conductivity which we should assign to a certain tissue type, and whether there's a strong variability between the conductivity of across patients or across subjects, for example. So there are really unknowns. Yeah, this is shown here. I mean, this is obvious. This is what I was talking about. But maybe this is a bit more interesting in the sense that this is CTs, low-dose CTs. That's why I don't see the brain. But you see the skull very nicely. And this is one subject which has a thinner skull, who has a thinner skull. Here you have a thicker skull. But what you have at the same time is when you look in here, this is much darker. So the density here is very different compared to here. And it's very unlikely that when you have strong differences in density, that you then have the same ohmic conductivity for the tissues here. But that's what we at the moment still model, right? We say that skull, we put the same conductivity there. So we can assume or we can expect that there are quite strong differences across subjects still, which we so far don't take into account. So how to validate it? That's actually that's more difficult. You can do it using invasive recordings. What was done in this study here is that they placed TDCS electrodes on the scalp. That's not shown here. Maybe they were here and here, for example. And because there were patients with electrodes implanted for preparation of epilepsy surgery, you could then also record the electric fields inside the brain created by the TDCS electrodes which were placed on the scalp. And when you do that, then you can go one step further. You can also create, again, head models of the subjects. You can calculate the electric fields at those positions, and you can compare both. By that, you have a validation of the models. The outcome here is that, basically it's shown here. Well, for some subjects, the fit seems to be quite good, with an R of 0.85 or so. That's quite okay. But we also have quite a few subjects where the fit wasn't so good. And here we don't have any clue why, right? So it was still that the head models were done in the best available or best possible way. They were all, for example, here in that study, they were semi-automatically done. So you look through all the slices and you correct. But still, for maybe half of them, the results are not so great. The problem here is that once you have that, you're a bit stuck in the sense, yes, you have now seen that, but there are patients. You cannot go in and say, yeah, let's do a little bit of variations, let's do experiments and see to figure out what's going on there. It's a one-time recording, then you're done. So we thought maybe we can come up with a method where we can do a similar thing, but in a non-invasive way. And this would be then MRI and so-called MR current density imaging. That's what we've been working for a long time on now to make, it's really for model validation. How it works is that, well, I talked about these two things here so far. When you have a head model or a head in general, and you place electrodes, it will create a current flow through the head. And when you have a current flow, this also creates a magnetic field. So this would be the current-induced magnetic field distribution. One component of it, where you see that that's a Z component, where you would say, okay, the magnetic field, red would go in that direction and blue would go in that direction. It's not so relevant. The point is that this you cannot measure. The current flow you cannot measure with MR, but you can measure the magnetic field because MR is working with the magnetic field itself, and it's very sensitive to changes of that magnetic field. And the currents here, this current induced magnetic field changes the magnetic field of the MR scanner in a systematic way, and that's something you can then aim to measure. The principle is actually quite simple. So you can again start with your current source, and what you do inside the MR is that you lead the current one time in one direction through the head, then switch it around, you lead it the other way through the head. You get two magnetic field patterns which are just opposite to each other, which are encoded mostly in the phase information of the MR images. You take the difference, and then you get an image which is proportional to the current induced magnetic field, and it's relatively easy to then also quantify how strong it is. Once you're there, you then can try to go on and use that information to, you have an inverse problem there again, to go from what you have there to how the current flow actually was. But at least you have a quantitative measure of at this level. The problem for those of you who do MR, you might have figured it out. We are talking here about a nanotesla range. This is really tiny. So this means that we put a person into a 3 Tesla in MR, and we try to measure changes of that 3 Tesla by maximally 1 nanotesla up and down. So that's not a lot, but it actually works. That's what we have been working on for some time. I don't go over the details now. I'd rather now go on and try to convince you that it really works. One thing, this was done by Frohe Gregersen then, one thing you quickly then notice, well, actually it's not only the magnetic field created by the current flow inside the head, which you're measuring. So you have an electrode here, you have an electrode here, you have a current flow through the head, and that's what you aim to measure. But actually what happens is that, well, when you have cables close to that, they also create the magnetic field, and it's overlaying. And when you now do this one here, this montage here with the cables here, and you just use Biot-Savart law to calculate the magnetic field of those cables only, then it looks like that, and in this slice here. And when we do the measurement, it looks pretty much similar, which means that when we don't control for this effect, we mostly measure the currents inside the cables and not the currents which we're interested in. That's a problem. We solved it in 2 ways. First of all, we optimized our electrode cable path. So instead of having them close to the head and bending, we now lead them down like that. And when you then do the calculation of the magnetic field in again the slice, it's much weaker. It's actually still there, but it's much weaker. And when you then measure it, you just see a very strong difference, meaning that what you measure here now is probably much closer to the real current-induced magnetic field. And the rest, which you don't see here, we can still take care of it by just calculating it because we know where the currents were. and applying Biot-Savart law and then subtracting it from our measurements. So with these two things together, we can get to the real current-induced magnetic field which we're caring about. Now, why do I talk so long about an artifact in a way which we can take care about? Actually, it also has a big advantage. We can use it to validate our measurements. So instead of having a current flow through the head, we can just have a wire loop around the head or here around the phantom, where we then can use Biot-Savart law to exactly calculate what we expect in terms of the magnetic field. And then we can just see whether we measured this also. And that would be an example here. So we have here of a head, we have the measured magnetic field for a wire loop around the head. And we have then calculated the expected or the magnetic field based on Biot-Savart law, and these are the residuals. And what we now care about is really how strong are our residuals compared to what we measure. This is here coded for such a dot pattern here, or this line here, and you see on average it's actually quite nice. Yeah. So by that we can convince ourselves that what we measure are really what we should measure. And that is really important is just shown here, where we during development saw some MR, whatever, changes in the MR sequence which we used. I don't go into details now, but it clearly turned out that we had a problem here where all the CSF was lighting up, which we then could solve. So it's really a quite nice test to ensure that what we measure is really then the current induced magnetic field of the currents flowing through the head. In terms of where we are now, is that we can measure 3 slices in 10 minutes with a really very good sensitivity of around 70 picotesla. That's in a stable way. I think that's for us now good enough to get started with using those measurements to really compare the measurements to the simulations. Now, after this technical detour, coming back to how can we use that to compare our volume conductor models to now what we have here available as ground truth? Well, one very simple thing is that we just simulate the current-induced magnetic field. So we go one step further. After we have simulated the electric field with the volume conductor model, we then apply Biot-Savart law to also get the magnetic field through the volume conductor model, and we can compare the simulations to our recording. And here it's actually not so bad. We start seeing nice correspondence. For example, we start seeing here these things popping up, which is due to that the ventricles shape the current flow a little bit. That's what we are seeing here. And on average, we have a relatively good correspondence with an R square of 82. That's okay. Let's do a very simple change. Namely, let's change the electrode positions from left to right. And suddenly our fit is much less good. And because we now did a lot of work to convince ourselves that we trust our measurements, the conclusion there then has to be, well, then our simulations are not that good for this kind of montage. Yeah. So this is just, first of all, the first possibility to, in a non-invasive way, validate how good our simulations actually are. And when we're at that stage, obviously, we want to take it further now to say, well, now let's try to use this information to update our head models to make them better so that they also work better in this way. What we started doing there is basically not changing the anatomy because they looked good, but we started to play around with the conductivities in a systematic way by optimizing them. I don't want to go too much into detail, but basically just pointing out that this would be the measured current-induced magnetic field, and by optimizing the conductivities, you can get apparently quite close with your simulated magnetic field to the measured one. So that's seems that okay, maybe going optimizing the conductivities is not the worst. Is a good possibility here. In terms of what it means in for our electric currents or which we induce, there are clear differences. So this would be a simulation based on the literature values, And this would be now the same simulation based on our optimized conductivities in the subject. And there's a relatively clear difference on the current densities also in gray matter. So it would matter for our conclusion how strongly we stimulate. This is just proof of concept. Quite interesting, but it has a big caveat, namely that it's really maximal overfitting what we did here. We acquired one slice with one current electrode montage, and we optimized the conductivities. And I'm pretty sure that indeed those current flow which we estimated here is closer to the ground truth than this one for this particular montage, for this subject, for this measurement position. But we have no clue how well it generalizes. Yeah. So this is only the starting point, or has to be the starting point. What we started now doing is to say, well, can we actually ask a slightly different question? Can we say, can we find a common set of conductivities which consistently improves our fit to our measurements, both for anterior-posterior montage and also left-right montage at the same time in several subjects. So there, obviously, it will not improve the fit so much, but does it work at all? And it seems to work in 4 out of 5 subjects. One is not really affected, but changing the conductivities to a new set consistently improve the fit. So, and by that we can then hopefully, when we take that further and just do more positions and more subjects, a larger coverage, not only one slice, but more slices, we can get hopefully to quite robust conclusions on what the best conductivity values are to improve. But that's still work in progress. So now, after having talked a lot about what volume conductor models are, how we can create them, that anatomy matters, and how we can validate them, I was going to spend the last 20 minutes also on applications. And here I'll start with electric stimulation, because that's my own research topic. And this is transcranial electric stimulation. The first thing is really just to ask the very simple question: when we put 2 electrodes somewhere on the head, how does the current flow actually look like? This is a pretty straightforward question, but it's something which people didn't do for a long time. And I just want to give you one example here, which is, this is the so-called F3, F4 montage, and people doing EEG, for them it's clear, it's just because we placed one electrode over F3, F4, and one over F3. And when you use that using TDCS, then you try to target the dorsolateral prefrontal cortices, which are maybe around here and here. And then you try to, for example, improve working memory, or also try to treat depression by that montage. So that would be the typical TDCS depression treatment montage. And people then write in their papers, we used F3, F4 montage to target the left DLPFC, for example, to treat depression. When you now run just a single simulation on this head model, then you start wondering if that is really true what they're writing. Because what they target is indeed the left DLPFC, but they also target all kinds of other structures, and actually with quite strong currents, also midline structures, which are also important, are known to be involved in depression, for example. So this just is a very straightforward tool to generate or test your hypothesis of whether the spatial targeting which you assume to have is really in place, or maybe whether you also hit other areas which are relevant for the task you try to test, or also the disease you want to treat at the same time, in addition to your original target position, right? So that's a very straightforward thing. And now, luckily, what has happened in the last 2 years is that when people want to now publish a TES, a transcranial electrical stimulation paper, it's now a good habit to include such a picture just to generally show, okay, in general, it seems that my current flow is as I expected and really focus on the areas which I claim it focuses on. Yeah. So that was a big change, very obvious thing, but it helps quite a bit. Then you can also just start comparing groups of subjects. That would be more or less, yeah, almost the same montage. Again, a frontal montage here in young and old subjects, and please note that the values here, so the color bars are quite different. It just shows that when you have old subjects and you apply the same montage with the same current, less reaches the brain. And this is just because there's more CSF now, so more of the current flows through CSF. Meaning that when you want to stimulate the old subject on average with the same intensity in terms of field strength in the brain, you should probably upregulate your currents a bit. In addition, we expect that there are also physiological changes going on at the same time, but at least on the physical or biophysical level, it's a relatively straightforward thing which we can actually account for, right? So we can start to use that information to make the treatment across subject groups more similar or control it better. Then, once we have volume conductor models in place, we can also start asking a very obvious question, namely, is actually the strength of the currents which we induce somehow linked to our physiological effects? I mean, it should be, but it's because TDCS is, or electric stimulation, is relatively noisy in terms of physiological outcome. It's not the most easy question to ask. So, but in a way, it's about determining really then what we call dose-response relationships, namely what kind of treatment dose do we need to get a certain response. And these are two studies which did that, both targeting with a standard montage, the motor cortex. And as you see, well, you target the motor cortex with this montage, but you also target all kinds of other areas. But for now, let's focus on the motor cortex, where you see that actually there's a large variability across subjects, how strong the field then there is. Some have a stronger field locally in the motor cortex, some less so. And then you can ask the question using magnetic resonance spectroscopy by looking at GABA changes. Is actually the change of the GABA level depending on how strong the field is in the individual subjects. And there were two studies. One did a current flow in a nodal direction, meaning from here till here, to excite the cortex. And there you see a negative correlation, and inhibitory you see a positive correlation. So that's quite promising. There are also other studies showing non-findings for other areas where we still have to understand why that happens. It could be the head models. They're maybe not so good for the other areas. But it's showing that it's going in the right direction. We can demonstrate dose response relationships. A very different dose response relationship is shown here. This is ECT, electroconvulsive therapy, that works for highly depressed patients as its last resort, where you then anesthetize the subjects to induce currents which are strong enough to really induce a short-term seizure. And the seizure works then as antidepressant. And it's very effective, so that's why it's used in clinic. But it comes with side effects as well, such as a short-term memory loss, for example. But the question is really, when you do that, can you then demonstrate some kind of a link between how the brain reacts to the ECT treatment and how strong the electric field was? Because at the moment, how this treatment is done is it's the same dose more or less for each subject. Nobody really controls how strong the field is. People can control how long it's given, because it's given long enough to induce the seizure, and then it's turned off, but not how strong it is. And the interesting finding here was that, yes, there seems to be a correlation between how strong the field was in the hippocampus and how strong the growth of the hippocampal volume was. That's one thing, which is then potentially related to the treatment effect. But at the same time, there was a negative correlation. The stronger the electric field was, the stronger also the side effects were. That's quite interesting. But it's also meaning that maybe we can take this to figure out like optimal dose, which has most likely a good treatment effect while avoiding side effects as good as possible. And then in the future, also individualize the strength of the treatments instead of just going to a certain fixed setting here to also individualize that. And that's, for example, work done at NIH, where they try to optimize now also ECT treatment in a very systematic way. And that's dosing is part of that. That's another dose-response relationship for TMS. So slowly people have the tool now available. As you had fMRI, now you can do field calculations, and people just do that and see whether they can find those response relationships. So I think that's really good and highly needed. So what was done here is that they looked at Parkinson's disease patients which had levodopa-induced dyskinesis. And so basically, when you don't give levodopa, this dyskinesis go away, and you give levodopa, and you have a certain amount of time you have to wait until they get the side effects of the medication again. And the question here was, when we stimulate a brain region involved in motor control, pre-SMA, with an inhibitory TMS protocol. They had no medication. You give medication, then you apply the rTMS protocol, and you just see how long it takes until they get the dyskinesis again. The first question was, is stimulation over the pre-SMA actually effective in prolonging the time until they get the side effects? And it was. And the second question was, is there a dose-response relationship? So those people who got a stronger stimulation in the pre-SMA with TMS, did they also have a longer treatment effect or effect of rTMS until they got the dyskinesia again? And there seems to be a positive correlation, which is now also used by a colleague of mine as a starting point for a larger study to say, well, maybe we can use that to individualize the dose in a treatment trial, which doesn't try to treat Parkinson, but tries to treat the side effects of the Parkinson's treatment. So I talked a bit about those response relationships, where we can use volume conductor models as a new tool to explore those. We can obviously also try to turn the problem around, because at the moment I was always talking about montages which looked like that, and they had quite unfocal fields, and then I told you, well, this is actually a motor cortex montage which tried to target this part of the area of the cortex, even though they create currents which are flowing in half of the brain. And the reason it was created like that was initially by pure intuition of the experimentators. So they said, okay, I want to stimulate the motor cortex, so I place one electrode over the motor cortex and one somehow far away, and then hopefully currents will go into the motor cortex. And then they, for example, used here TMS as a readout initially to say, well, when I do that, I see inhibitory or excitatory effects on the motor cortex. That's how they did the proof. And yes, it works, but obviously you don't have to stop there. You can also turn the question around and say, yeah, that's fine that you have such a montage, but maybe we can get better than that and only target the motor cortex. And that's an optimization problem. When you allow for multiple channels, so that's now looking a little bit like an EEG cap, but the idea is that you can now induce currents in each of the electrodes, and you try to superimpose those currents in the optimal way to only hit the area which you want to hit. And that's an optimization problem which is related to EEG inverse solutions, but it's not identical, because what you have is still the safety limit. In EEG, you don't care about it, but here you have hard constraints on how much current you can induce into each of the channels, for example. I don't go into the detail, but so people liking math will like that. Others, well, not so much, but we like working on these things. So that would be then the outcome of a corresponding multi-channel montage, where we use the outcome or the field in the motor cortex created by this montage, gave it as a target to our optimization algorithm. And as you see, now you get the same field in your motor cortex region, but it's just much more focal, right? So we can now focus really with optimization, targeting optimization. You can focus your field really on the area where you want to have it. That's made. Does it always work perfectly? Well, let's see. I just have one example where we said, okay, let's just make up this montage here, which probably would target some SMA a little bit deeper down. So we extracted the electric field there, gave it as a target to our optimization algorithm, and that's what the algorithm came up with. Now, when you compare this field to this field, you would say, yeah, it's much more focal, but it's by far not really focused on the area. And the reason is just that this is a deeper area, and by pure physics of the current flow, you cannot magically focus currents going into deeper areas and focusing there. That's just a limitation. So basically, it just means that all our targeting optimization works fine for superficial areas. It helps also for deeper areas, but to a far lesser extent. That's just a limitation in general. We are always quite good in transcranial stimulation techniques, except for ultrasound, which I don't talk about today. We are quite good in focusing, being relatively focal on superficial areas, but we are less good in when we go to deeper areas. When we have electric field simulations around, we can also apply them to redo or improve protocols for TMS, which have been used for a long time, which is so-called motor mapping. What you do there is you stimulate at many positions close to the motor area. You always record the responses. And you just here record via a heat map here, for example, how strong the responses have been. Blue means no twitch. Yellow means, yeah, there was a strong twitch. Then there's orange, stronger twitch. And red was probably then the strongest twitches. You then have a lot of recordings on this 2D grid. And what people did so far now is that they just said, well, let's calculate a center of gravity of the recordings. This gives you some position on the skin surface, because what is shown here is just the position of the TMS call, not the field. And basically they're saying, yeah, on average, we get the strongest response when the call is placed here. And then they concluded from this call position on where they stimulated in the brain, namely simply the closest cortical position. And this is a problem, which is, for example, shown here when you end up at such a position. This is all gray matter here, stimulated by TMS. So you have no clue on when your TMS call works best at this position to induce a muscle twitch, whether you stimulate the cortex here, here, or here, right? You have to make assumptions. So, but when you have electric field simulation in place, you can turn the problem, or you can reformulate the problem a bit differently. And the idea here is really that we try to search. Well, first of all, for each position which we record, we have the muscle twitch, but we can also estimate the electric field. So what we do here is we try to link both things together. We just try to see at which position on the cortex the electric field changes correlate best with the changes in the muscle twitches. So we just, because when you change the position of the call, you get also at each position on the cortex, you get a slightly different electric field, and you get a slightly different muscle twitch. And you basically can just try to correlate both things to see where the correlation is best. Usually we then do segmental fits, for example, And where you then would see this is basically then a position where it worked best, where you have a nice link between the electric field and the MEP on this side, right? And it's then leveling off when you go away. That also tells you something about the focality of your stimulation. You not only learn where you stimulate most likely, you also learn something about those response relationships again at the same time, which is also a nice thing. And the other, finally, good thing is that instead of having it to do on a grid, which you try to make as similar as possible, so or equally spaced as possible, here the procedure is actually quite nice for the experimentator, because on purpose, you try to just randomly sample positions on the head with different orientations, which is quite comfortable, right? So you don't have to accurately target. That's something, your position is recorded. It doesn't matter where you really are. You just put your call on different parts of the head, you sample for some time, and then you let the computer do the work later on for you. So also from a point of application, it's also easier actually. Lastly, I want to go to EEG source localization again, because I mean the problems are quite linked. So then you have volume conductor models. Then you can also try to not only calculate, estimate the forward problem in the sense that you have an electric field, or you have a current source somewhere in the brain. You calculate how the distribution on the surface of the skin looks like. But you try to solve the inverse problem. That's what you have usually. You have a recording on the skin surface at your sensor positions, and you try to estimate again where the sources are which created this recording. So there are several algorithms out. I don't want to go into details. What Jesper Nielsen did here, he just then looked into how important it is again that the volume conductor models are accurate, because I showed you that at least for the forward problem, it makes a big difference. Here, actually, it was surprising to see that for the inverse problem, volume conductor models seem to matter less. Which is just pointing to the fact that inverse problems are so highly regularized to work at all in EEG that some parts of the changes are not in the forward models are actually not so relevant anymore. So what is shown here is different types of inverse solvers, dipole, music, S. Loretta, for example. What is shown here is the SNR of our simulated EEG. And here would be then the localization error. In general, you see clear differences between the methods for a single source, I have to say. You see clear differences between the methods. Some are just more unfocal compared to others. But then when you look into how strong the head models or the accuracy of the volume conductor model influences the solution, then first of all, you see that it seems to be more relevant when you have a higher SNR measurement, which somehow makes sense. And then it also seems to make more a difference or more effect on the more focal algorithms. So for example, for the SPM, there's really not much difference between the head models. It doesn't matter which one you put in. You always get the same peak localization errors. But for more focal montages, such as music or not montages algorithm, it seems to matter, and there again, the more accurate head models seem to help, which makes sense. But it's actually not that massive. This would be now an example for dipole sources, for dipole fitting, and you have to look a bit more closely to see really the difference between the different head models. For example, here, SPM-based segmentation and field trip, they don't seem to be doing so well for the temporal poles here. The other methods seem to be better. When you go, maybe here in these deeper regions in general, you see that the localization errors are a little bit higher compared to here. But it's actually relatively moderate. We were surprised by that. We want to follow up on that now a bit more in testing whether this is also the case when we have more than one source. This is the most simple case we have, but whether how much it really matters when we, for source demixing, when we have more accurate volume conductor information. And with that, I want to conclude my talk. Mostly I was talking about volume conductor models. I tried to convince you that they matter in terms of personalizing them. So when we have that the individual anatomy really determines how the electric fields look like, which we induce by TMS or electric stimulation, or which we record by EEG. I talked a little bit about MRCDI, so our attempts to really improve volume conductor models. And then I went through a few example applications to use them, mostly in TMS and TES. So for brain stimulation, where we can use them, first of all, to characterize how our stimulation actually looks like on the brain level, and then also try to improve it, first of all, by systematically optimizing them, but also by really learning about dose response relationships, which is really something which is so far not well estimated or well investigated, in the sense that when you have a certain target in the brain, I cannot tell you, okay, you have to target this value, this area now with 0.5 volts per meter with your electric stimulation for 10 minutes to get the best effect. That's information which is still missing. In order to build up this information, we can now use this systematic dosing studies to actually explore just on a very experimental level how dose is related to the physiological outcome. Yes, and with that, I'd like to stop, and I'd like to thank all the people doing the work, and partly they are now in my group still, or they moved on, but they were all instrumental in building up the methods. And with that, I'd like to thank for your attention.
Okay, so any questions from the audience?
Yeah, thanks for the very nice talk. I'm wondering, when you do this stimulation, do we know what changes at the neural level? Like, do we have more synapses that are created, or do we have, what do we know about changes? You showed like GABA changes maybe, and also to continue the question, do we know other like receptors that change?
So that's really interesting, but we know surprisingly little about that. There's a ton of data on the human motor cortex. The reason is that you can then always ask the question, how did your intervention change the muscle twitch strength? And there are very elaborate experiments out which try to make then a link between your GABA levels and so on. And you can also combine that with drugs to block certain channels to a certain extent. I mean, it's humans, it's not animals. You can give a drug which then modulates slightly. And again, so that's where we have our knowledge from, so that we can tell what kind of, whether GABA and glutamate and so on plays the role in building up a neuroplastic effect. But then the next step, and really to say, okay, this is the mechanism behind it, and this is how much synaptic changes we see. No, this is still an open field, which is surprising because the depression treatment really is now gaining a lot of momentum after 25 years of development, or even more. But on the very fundamental level, we are really still pretty weak.
There's no animal studies.
No, that's really surprising. I mean, there have been animal studies initially for safety, and that's fine. But in terms of mechanisms, we are now finally starting to go to more the mechanisms in two directions. The one is I was only talking about the electric field, but in a way we don't care. We care about the neural effects which the electric field creates. So one thing is to start combining electric field models with neural models. That's something we are now working on, to really just ask with very elaborate, fine, highly structured or highly detailed models to just ask the question, what does this field do to this neuron? And then on the other hand, also people start looking more into animal studies to really try to figure out, okay, what does the intervention really do? And I think we have to go from these two sides and to meet somewhere in the middle, hopefully. Yeah.
I have a question on MRT CDI.
Yes.
What you showed is that the impedance is anisotropic, what I understood.
Yes. So we model them in the moment. At the moment, we model them as isotropic.
Yeah. So then my question is, if there are studies like ex vivo, like you know more in detail that for example, in some regions of the brain, then anisotropy is this way, and then in other regions this other way.
Mm-hmm.
And also, if you have like ways to establish or to measure this anisotropy indirectly, like maybe with diffusion MRI or something.
Mm-hmm. Yeah, exactly. So there's not so much. There's a little bit ex vivo knowledge out, and indeed, conductivity is anisotropic, especially in white matter. What we mostly did is we did sensitivity analysis by informing our models, for example, by exactly what you said, by diffusion tensor imaging and making some assumptions on how diffusion anisotropy maps to conductivity anisotropy. And then ask the question, how much does it change the fields which we estimate? So it's a pure sensitivity analysis. And there the changes were always moderate, in the sense that other factors, they are there for sure, but other factors seem to matter more, such as the thickness of the CSF layer, because that's very well conductive, or how you exactly model the skull. So yes, it's a relevant factor, but maybe not the most relevant at the moment. So that's why we at the moment still fit our MRCDI data to isotropic models, because we don't think that using anisotropic models would give us a big benefit there.
I actually had a question a little bit related to that. I mean, are there any other MR modalities that you would be interested that you try to incorporate into this to better estimate the conduct? Here's at various sites for, I mean, for example, just proton density imaging, was that be correlated to the conductivities, for example, or
potentially
other yeah, or other.
So I think one thing is that the conductivity of the skull plays a big role, and their MR is just really bad. Otherwise, yes, you can think of potential correlations. The question is then always how to prove such a correlation. Also, like what between diffusion tensor images and or anisotropy and ohmic anisotropy, there should be a link. Most likely, how it is exactly, that's difficult to establish without invasive studies again.
Thanks for your talk. I have a question about the intervention of TMS. During the intervention TMS, when you're using the EEG to monitor the effect, and after the intervention of TMS, there suddenly have immediately fluctuation of the signal, and then how you process the artifact or noise on the EEG signal.
So you're specifically asking about TMS EEG online?
Oh, yeah.
Okay. And there's this artifact after TMS period, that's correct. And I think you can get quite good signal after some time for most central brain regions here, where you don't have the strong muscle artifacts. So when you go further laterally, you will just have extremely strong muscle twitches, which interfere a lot with your EEG recordings. When you go more centrally, I think this is less of a problem, and you get a clean EEG signal after 10, 15 milliseconds when you do it the right way. The larger challenge is then that TMS also has auditory side effects and somatosensory side effects. You hear the click and you feel it. So even though you have a clean response to record, you still record a mixture of different sources. And what you're really interested in is only how TMS acts on your, not only, but on the network where you stimulate a node, right? And then it spreads to within a network, and that's what you want to track. But because it's overlaid by a lot of auditory co-activations and somatosensory co-activation, that's really a tricky thing to do. Yeah. So that's why people now shifted to the more early components of the EEG response. Then the question is, how early do you have to go to really believe that what you record now is really your primary TMS response and not some overlaid with some other co-activations.